Hello guys. Hi everyone. So today I'm going to discuss about Nobel Prize in Physiology 2024. So it is about the discovery of microRNA and its role in post-transcriptional gene regulation. So here what I'm going to explain is how this discovery is being made and what is it and how it impact a common people, or you can say that a layman person, how it impact us in terms of our scientific progress for human race as well as understanding biology. So this discovery was made by two scientists. Their name is here you can see that Victor Ambros and Gary Ruvkun. So they were they were the pioneer in discovering them for the first time, the microRNA, you can say, and how it impact how it affect the gene regulation. So let's start. I think currently these people are in MIT, so I don't know the current status. You can also look into the Google their current positions, and one is from Harvard. Okay, both of them are from MIT. I think yeah, I'm from MIT. So let's move on to the explanation. So what is this microRNA and what does post-transcriptional gene regulation mean? So if you talk about our human body or any multicellular organism like rat or let's say any parasite, or you can say that maybe any mammals or plants, you can say. So you see that there are different type of cells, or you can say at least that there are different organs in an multicellular organism, and these organs are actually made up of even the whole body is made up of cells, and these cells are of different type, you can say, and they have different functions. Like if you talk about liver, then our liver have different functions and our lungs have different functions. So it is decided by the type of cells that is present in that organ. And even though the DNA, that is the genetic code that is present inside our cells is exactly same in all the cells, but the cells behave differently. So scientists were very much curious from very early time that how does the same DNA, even though they have the same genetic code, you can say that the same code in all the cells, the cell behave differently, and it is able to look different as well as perform the function differently, and also responds differently when excited or when given some stimuli. Okay, so for a long time it was not known how this why this thing happened, and people were mostly thinking that the scientists were mostly thinking that protein is the ultimate thing, that actually obviously protein is the ultimate molecules that actually provide the functions it have. Like let's take an example of insulin. So if you talk about insulin, then insulin is able to regulate glucose, right, in our blood when we eat, and insulin is a protein. So this insulin is secreted by our pancreas, and it cannot be secreted by any other cells like blood cells, or you can say the heart cells. So even though the same DNA is present in the pancreas cells as well as the heart cells. So scientists thought that okay, there are thousands of protein that are being manufactured in our cells, and that is somehow controlling the function of the cells. But why different proteins are being expressed, or let's say produced, in different cells? Why not insulin is being produced in the liver cells? Why not it is produced in the heart cells? So this was a very challenging questions, and they wanted to understand. So the two scientists that I mentioned were able to discover you can see here that they were able to discover a microRNA. So this is a type of RNA, you can say, and that is somehow regulate. It's a type of regulation. There are multiple different type of regulation, but this is a type of, you can say, a type of regulation, or you can say that a type of controlling the behavior of the cells. So this microRNA is able to regulate which protein will get expressed in which cells. So there are different type of microRNAs, and I think around 10,000 microRNA till now it has been discovered. And they discovered this microRNA in an organism which is called C. elegans. It's a parasitic type of, it's a worm, and generally you can see this is actually the worm. And they discovered that one microRNA is able to control the expression or production of different proteins. Okay. So, but before that, I want to explain that to produce a protein, first what happens is that this is very basic in molecular biology that we study, that is from DNA. The first translation of the genetic code happens in the form of mRNA. So this is a molecules which is being produced from a DNA molecules, or from a part of the DNA molecules, and this is called transcription. And then it move on to the protein part, where mRNA is being translated into a protein or chain of amino acids, and which is able to do certain functions, just like insulin, what I explained before. So this is called central dogma, and the protein it produced is able to do the functions. Now if you come back to this thing, so you can see that they have discovered this microRNA which is able to regulate the expression of the expression of a DNA molecules, somehow inhibiting or controlling the expression of the mRNA of that gene. So they discovered this, but many I think it was like many scientists were not so intrigued by the discovery, because they thought like it's what's so unique about this, it is discovered in a worm, and that is not somehow related to human being. But later on they also discovered another microRNA which is actually present in human being, and they were able to show that this type of regulation or this type of controlling mechanism is present in most of the multicellular organism, and it is very much important in how in understanding how our cell behave differently, even though they have the same genetic code. So this help us to understand a type of controlling mechanism in our body that is responsible for different cells to behave differently. Now why this is important? Because there are various diseases that happen to human, and we try to understand them through different experiments and different ways. And it has been seen that in different diseases, this microRNA are also being differently. They also express differently. They are also produced differently, or they behave abnormally and somehow lead to the disease condition. So because this is very complicated, and not only this, but there are several level of regulation is present in the in a multicellular organism, but this is one such way of regulation. So this discovery is very much important in understanding various human diseases and how a multicellular organism control its different cell behavior and able to function properly with different organs and produce a multicellular organism or a complex organism. So this is I hope you understood, and this is very much important in our development, in our physiology, in disease and cancer. So everywhere this discovery is impacting this. You have this understanding of how this whole biology happen in a multicellular organism, and from a human perspective it is very important. And now from a research point of view, I want to also add here that it is being actively used in research, and here I think we don't have an example, but because in research what happen is you want to understand that some DNA section, or you can say some part of the DNA, let's say this blue part, what does it do, or let's say what does its functions, what is its functions. So because of this understanding, we were able to artificially produce a microRNA system which is able to inhibit the protein production. So in research it is being commonly used to exploit this system to understand the behavior, understand the function of that DNA sequence or mRNA sequence, that what does it do in the cell, or what does it do in an organism. So let's say you want to understand what does insulin do, and you have injected a microRNA that is blocking the production of the protein, and that is insulin. So you have because that are very specific to insulin mRNAs, and you have injected the microRNA and it is able to inhibit the production of insulin, and then you see what happens to the animals, or let's say human, because we cannot do experiments in human. This is generally done in mice or any other organism which somehow mimic the human physiology, and then you observe the human organism, that what happens to that organism, and you see that there is imbalance in glucose metabolism, or very high rise, very much spike in the glucose of the blood. And due to that there are various malfunctioning of different organs, and all those things happen. So that's how it is being used in research, and it is also leading to lot of discoveries in research and our understanding of the biology. So I hope you were able to understand what I'm explaining, and in future I'm also going to bring a lot of other videos like this explaining different concepts in biology and in advanced biology, let's say. And these are the key publications that they have made for the first time that shows this discovery. You can go through it, and this is the documents that is published by the Nobel Prize committee. So you can read and understand if you want to go through it. Thank you.